Good afternoon, class. How are you guys today?
Good. How are you?
I am doing great, and we're going to continue our studies of the different bacterium in nature. So our bacterium for this week is Deinococcus radiodurans. And let's first visit the characteristics of this bacterium. This bacterium is unlike other bacteriums that we studied in this class. So it likes every 4 D radiodurans likes to come together and form tetrane. Tetrane usually appears smooth and convex and appears in red to pink color. As we can see in this picture, the dark red indicates the genomes. The diameter of this bacterium ranges between 1.5 to 3.5 micrometer. Also, this bacterium has a gram-positive stain. So in this class, we studied that there's 2 types of stains, gram-positive and gram-negative stain. Gram-positive means that the cell wall is composed of a thick layer called peptidoglycan. However, in this bacterium, peptidoglycan, it lacks from L-ornithines and teichoic acid. Also, the classification of this bacterium is Deinococcus, and the phylum is Deinococcus thermus. So this bacterium was found in a meat can. This meat can was exposed through a lot of radiations, and in order to sterilize it and store meat. However, the meat kept getting spoiled. So the scientists wanted to do a research over how and why the meat was getting spoiled. So this bacterium was found in the can in 1956. This bacterium is known for its major characteristic of radiation resistance. Therefore, it gained its world toughest bacterium. The world toughest bacterium title in the World Guinness Book. The radiodurans, as I mentioned, it's capable to survive through extreme environments. That means it can survive through cold temperature, high temperature, acidic environments, which give it the ability to survive on the ground, nuclear reactors, and on top of spaceships. So this bacterium has the capability of taking in foreign DNA and incorporate it in its own genome, which makes it a perfect species for research of bioremediation. Also, it is able to efficiently detoxify radionuclides and other toxins in more stable compounds. So, the growth of D radioduran is accomplished by 5 minimal sources. First, we have carbon, nitrogen, sulfur, manganese, and we have nicotinic acid. The bacterium way of producing energy is unlike other bacterium in nature. It uses a vacuolar type of proton ATP synthase instead of F0, F1, like we see in the picture over here. Also, D radiodurans carries the characteristic of thermophiles by encoding multi-subunit Na+ and H+ antiporter. The bacterium genome encodes functional pathways of glycolysis and tricarboxylic acid. Also, D radiodurans uses amino acids as a nitrogen source instead of using ammonia as a nitrogen source. It can use a variety of sugars such as carbon for energy source as well. The 4 stages of the life cycle of this bacterium are lag phase, log phase, stationary phase, and death phase. So as we can see in the picture, let's have a much more clear idea of the life cycle of this bacterium. Lag phase means that the bacterium is not producing and there's no death rate as well. So log phase means that the percent of reproduction is higher than the percent of death rate of this bacterium. Stationary phase is when the reproduction equals the death rate and there's no change. Then the death phase shows a decline in the graph, which indicates that the percent of death of microorganism dying is higher than the one that they're producing. So for this class, I want you guys only to learn only 3 important proteins that aids for the survival of D. radiodurans. The 3 different proteins are redox hydrolase protein, acetyltransferase, and manganese. Redox hydrolase protein is the house cleaning enzyme. It destroys any potentially deleterious compound. It also prevents the incorporation of AOXO-GTP into DNA. Acetyltransferase enzyme job is to detoxify antibiotics and toxic products that arise upon DNA damage and regulatory protein acetylation. Manganese is an ion that prevents oxidative damage to repair protein and allow them to swing into action after radiation has damaged the DNA. That's one of the reasons why D. radiodurans can survive high radiations. There is all the time DNA repair enzymes that would fix the DNA to its normal function. D. radiodurans have not been associated with any diseases host, mutualism, communalism, so any of the symbiotic stuff that we covered before, this bacterium was not associated with them. So I'm sure by now everybody's wondering, why are we learning about this bacterium? So through our lecture, we learned that this bacterium has the capability of surviving through a lot of radiations, also the ability of surviving through really harsh environments and very poor nutritional environments. So it's a perfect species for nuclear waste pickup, also for medical uses, and for space traveling. So as we all know that space travelers always complain from radioactive sickness. So we can incorporate this bacterium way of survival from radiations into many ideas to fix this problem that our travelers experience while they're traveling through space. Also, it can survive through extreme radiation, the ability of repair its own DNA. So the main thing that I want to emphasize in this lecture, that this bacterium it has the capability of repairing its own enzyme. That means, like, no matter how much radiations it can get or consume. And it has the ability of getting back all of its DNA to work from the DNA repairing enzyme. So the DNA inside of this bacteria, the DNA has many copies of itself in its own genome. So it's capable to survive through these conditions. So any questions?
Are there any other uses for D-radiodurin?
One of the, if scientists really understand, if they go into understanding the mechanism of D-radiodurans, there is a possibility of using it in treating cancer. So as we all know that our cancer patients get exposed to a lot of radiations through chemotherapy. And one of the most important things is that our patients experience hair loss, a lot of other stuff. So if we can harness the thing that helps the radiodurin survive through these radiations and cooperate in non-cancerous cells in the human body, we can help these cancer patients not lose their hair and get affected by these type of radiations. So yeah. Any more questions?
No, that's it. Thank you.
No problem. And that's it.